So welcome everyone to another SOL seminar this week. Today I'm glad to introduce Aureliano. As a short biography, Aureliano Bombarely obtained his PhD in 2007 in the University of Malaga, working in the analysis of the molecular mechanisms of the strawberry ripening. He moved to the US in 2008 to work under the supervision of Dr. Lucas Muller in the Boyce Thompson Institute for 5 years. Then he switched from molecular biology and genetics to bioinformatics and genomics, working in the SOL genomic network database and the production of several genomes such as tomato, tobacco, and Nicotiana benthamiana. His exposure to the polyploid genomes made him to reconsider his career in bioinformatics and database development, and he moved to the laboratory of Professor Jeff Doyle at Cornell University in 2012. He worked in the elucidation of the origins of the glycine polyploid complex. In 2014, he obtained an assistant professor position at Virginia Tech with the opportunity of developing his own group. He worked in the development of several bioinformatic tools and genomes such as petunia and zucchini. At the end of 2018, was recruited by the Università degli Studi di Milano with a term position as Professore Associato to teach plant evolution. More research lines pop up at this time, including the genomes of mango, avocado, and pawpaw. He recently moved to his current position at the Instituto de Biología Molecular y Celular de Plantas in Valencia, Spain, as Professor de Investigación. And with that, today is going to present about insights of the genome evolution in the Nicotiana genus. And so we welcome Professor Aureliano, and you can start sharing your screen.
Thank you very much for the invitation and for the nice introduction. So let me see. I'm going to go to the slideshow. Okay, and then I can share it. Okay, can you see the screen?
Yes, perfect.
Perfect. Okay, so as I said, thank you very much for the opportunity to give some of the insights of some of the work that I have been doing. I will not say recently, because I have been linked to the benthamiana genome now for, let's say, more than 10 years. And I will talk a little bit about that, and also a little bit about some work that we have with some of our collaborators. Okay, so let me... Okay, so this is kind of the thing I'm going to try to talk today. Of course, I'm going to give you some introduction about the Nicotiana genus, one of my favorite Solanaceae, for all of you that you don't know it. And then I'm going to summarize a little bit about the Nicotiana benthamiana genome, what is the thing that we did. And then I'm going to talk a little bit about the work that we did on the elucidation of the origin of the Suaveolentes section. So let me start with a little bit about systematics. As you know, Nicotiana is a genus of herbaceous plants that can find in almost all the continents, America, Australia, South Africa, some Pacific, cannot be found naturally, for example, in Europe, but I have been working in some places, and sometimes I saw some Nicotiana plants that I think that they may be escapes. The diversification between Nicotiana and Solanum was estimated in around 24 million years ago. Okay, but this date, of course, changed depending of the work, but this is a good summary of that. And as you know, some of you know, included around 90 natural occurring species divided in 13 sections. And Nicotiana benthamiana, for example, is in the Suaveolentes section. I said 90 because, but 90 is now, maybe next year is going to be 95 because new species have been discovered last year. No, if we see the distribution of Nicotiana, as you can see here, as I said, you can find in Europe, but most of them are escapes, some of these things. But yeah, mostly it's in the Americas and also Australia and some in Africa. Of course, for example, I'm going to talk about the Suaveolentes clade, that is, you can see here in green. If you can see in blue, is also because you can have some escapes too, but naturally are these Suaveolentes species. And the genome sizes, and is how I'm starting to work with them, go from 1.35 from Nicotiana trigonophila to 6.35 of Suaveolentes. And the thing that is why I'm starting to work with them was because around 40% of them are polyploids. So it's an excellent model for studying polyploid and genome evolution. As I mentioned before, recent species of Nicotiana has been discovered. And I have to say, because I'm quite excited of those, so always to have new species in your genome. So these are two, Nicotiana grandalera and Nicotiana rupicola, that was recently published. And also, of course, this wonderful work of Marches and others on the Suaveolentes species, and they have a really new species. If you're having attended these online seminars, I think that they present some of them. So I like this slide because summarize not only some of the distribution of the Suaveolentes species, but also at some point starting to plot them not only by the species, but also based on the chromosome number. As you can see, the ones that are bigger are the ones that have more chromosomes, and the one that has smaller have smaller chromosomes. And that is important because also the ones that have more chromosomes are be thought that are the ancestral ones. I'm not going to talk a little bit. I'm not going to talk too much about that, but this is something interesting because also it can define how they're starting to colonize Australia. So there are some hypotheses about the origin of the Suaveolentes section. So that can be summarized here, and also was summarized by Stephen in his publication in 2020. They are derived from a single allotetraploid origin from South America, where most diploids are native. Also, they have a significant diversification post-polyploidization event and the process of polydiploidization. And most of the species of the Suaveolentes appears to be rising in the last 2 million years, although the origin of the section was 6 million years ago. And it's more or less the thing that I'm going to talk about the origin of these, not about the diversification, but about the origin. So there are several hypotheses about the origin of the Suaveolentes section. It started with Goldsmith, that he said there was an hybridization between Petunoides, Noctiflorae, and at that time, Alatae and Silvestris worked together as a section. Later on, James Clarkson starting to use some plastic markers that pointed out that the maternal donor probably was in the Noctiflorae section, and the paternal in the Alatae Silvestris section. Later on, with the nuclear gene GS, the glutamine synthetase, was pointed out that maybe one of the ancestors was trigonophila, but was not very well supported, and in some ways was very well discussed in that publication. Later on, Laura Kelly starting to add more genes, more nuclear genes to this analysis, pointing out that it was Noctiflorae Petunoides, the maternal donor, again, being the paternal donor Silvestris. And one of the last works analyzing the Nicotiana benthamiana genomes pointed out more or less the same thing. So that is kind of the hypothesis that we're starting to work with in this work. Also, there are several studies about the Suaveolentes phylogenies, not only based, one of them, for example, based in morphological characters, but also some excellent works by Stephen and Mark using different plastic markers, and then the whole chloroplast, and also most recently rustica data. I'm not going to talk about that, but also one thing that is interesting about the diversification was how they're starting to colonize. There are the two possible routes. One of them is, one of the Suaveolentes species is Nicotiana africana, that is the only the only Nicotiana section, or Suaveolentes section, sorry, in the African continent. And the other is, you have also some Suaveolentes accessions in the Pacific Islands, like Nicotiana fragans. So there are two possible routes from South America to arrive to Australia. One of them will be through Africa, and the other will be from the other side. Okay, so that is also other thing that is interesting about these things. Okay, and once they're starting to arrive, there are two possible points One of them, and based it also in the number of chromosomes, and based it also in the phylogenetic studies about what are the oldest species in the Suaveolentes section. And probably the thing that happened is that they starting to arrive here, that when you find this Nicotiana forsteri, or the Neji, they starting to arrive here, and maybe they're starting to colonize the rest of Australia. Okay, so that is kind of the background, the landscape. So I'm going to start to talk a little bit to introduce the genome that we have been working for several years now, that is the Nicotiana benthamiana genome. I started to work in that, as I mentioned before, more than 10 years ago. So we published a very, very bad draft at that time, but was very useful, I guess, in 2012, that was mostly based only in the Illumina sequencing technologies that was at that time. And since then, we have been improving this reference genome based in, let's say, data donations. No? So some people that we have been working on, maybe people that was interested to improve the genome, just were doing more sequencing and sending the sequence to us, and we have been trying to improve it. So over the genome that we have in 2012, we have added more libraries of Illumina. Also, we add some PacBio old sequencing that Professor Brian Kipko sent us, and also Celine Song sent us some high-C data that we use. So when we use everything together, no, we produce a first assembly with Illumina data, then we fill the scaffolds with PacBio, and then we did another rescaffolding of the context. And finally, we use this high-C data to do an extra round of scaffolding that was great because we were able to put more or less everything in chromosomes. No? So we ended with an assembly size, or with a genome with a total size of 3.303, that is more or less the estimated genome size. We were able to anchor more than 95% of this assembly into chromosomes, and still we have some small scaffolds. And as you can imagine, the longest sequence are chromosome size, that is 194 megabases. So here you can see more or less the distribution of the different chromosomes. So some of them are bigger, some of them are smaller. We tried to also to do some genetic map, but at that time it more or less failed. We didn't have enough markers to build a solid map. So we moved with a high-C. We did some evaluation about the quality of the genome. So we use Mercury that was published recently. And it can be estimated that the completeness of this genome is around 98%. So it's kind of okay. Remember that we use Illumina. So it's a base Illumina that we improve with PacBio. So that means that probably most of the centromeres and the telomeric regions that are highly repetitive are not contained here. Also, the quality value is high, also because it's done with Illumina. So, and the consensus rate is more moderate low, no? We can say. We did also some annotations with repeat modeler makers of this genome. And we ended up with around 61,000 genes. These 61,000 genes are more or less twice the number of the diploid genomes that we have as a reference, for example, Nicotiana attenuata or Solanum lycopersicum, and a little bit less than Nicotiana tabacum, that is a more recent polyploid. When we run Busco, we ended with around 96% of Busco completeness, and as you can see also, the duplicated genes are high, as expected, compared, for example, with a diploid Nicotiana like Nicotiana attenuata. In terms of repetitive elements, also this polyploid genome was starting to just purge some of the repetitive elements. So it has the percentage of the repetitive elements is lower than, for example, the genomes of Nicotiana sylvestris and tomentosiformis that we had at that time to compare, or for example, than Nicotiana tabacum. The thing that maybe is interesting is that if you compare with these genomes, you have less gypsy repeats, more copia repeats, just based in this genome assembly. And also based in this genome assembly, we did some syntenic analysis with the genome of Nicotiana tabacum. As you can see, we have a pretty good synteny for some of the chromosomes. No? For example, the chromosome 8 of Nicotiana benthamiana is the chromosome 4, and the chromosome 6 of the version 4.5 of Nicotiana tabacum. Okay, I have to say that of course, there is a new version of the Nicotiana genome that is being produced by the new Nicotiana consortium. I think that is available right now. And that goes down with PacBio, and the quality is even better than this one. Okay, so that is kind of talking a little bit about the Nicotiana genome that we have. We submitted the Nicotiana genome to the SGN database, so that will be available. I think that it should be available quite soon. So as I said, let me talk a little bit about the illustration of the origin of the Nicotiana section. So we have two aims when we're starting to propose this work. No, the first is, okay, so where the Suaveolentes section come from, and the second is if we could detect or identify patterns of the genome evolution associated to the process of diversification. Okay, just looking to this genome. For that, we did our collaborators did the resequencing of 16 Suaveolentes species and 2 subspecies using Illumina, okay, with at least 20x. I'm going to talk today only about the aim one, that is more or less the work that we have almost done and rounded. So we're starting to do some genome size estimations of all the different Nicotiana species, and of course some Nicotiana Suaveolentes. The thing that for me was interesting here is that in some of these accessions that we were measuring some genome size, we found some accessions that was, I mean, of course, Suaveolentes is a polyploid section, and you have polyploid species, but of course you can have some some internal duplications in some individuals, as we saw, for example, here. No? So we have some of them that were duplicated. No? So we found that the Suaveolentes sections goes for a genome size from 1.7 Nicotiana cavicola to even, well, at least according to our estimation, 7.3 that was the Nicotiana debnegi or Nicotiana forsteri. We found that several clades show some possible polyploid accessions. So we found Nicotiana excelsior with the expected genome size, and we found a different accession that has twice that size. We still am not sure why, but we repeated the measures and we found that, so we don't know if some of them can go for endoduplication in the lips or any other processes. And of course, we did some genome size studies associated with the chromosome number, and we didn't find any association. So for example, of course, the more chromosomes that you have, bigger is your genome. But then at some point also you have some accessions, some species that even they have 30 chromosomes, still they can have quite big genome sizes compared with, for example, to other ones. Of course, we did all this work just as a preliminary work before we started to do the sequencing to more or less know how much we should start to sequence. Okay. So this is the list of the accessions that were sequenced by our collaborators, as you can see, from Nicotiana africana to Nicotiana velutina. For some of them, even we sequenced 2 different accessions, and we try to hit at least I would say at least 40x of coverage for each of those. That for some accessions that, of course, they have big genomes that can be allowed. We also, of course, because we wanted to put everything in context, we also downloaded all the public assembled genomes that we have at that time, and also some scheme data, and also all the sequencing transcriptomes that you can see here. In green are the ones that you have the genome sequence, so we didn't do these assemblies. But for the ones that we didn't have the genome sequencing, so for example, for Nicotiana linearis, we did the transcriptome assembly of those, trying to just hunt as many genes as we can, okay, for our studies. And as I remember, our first aim was trying to put everything in the phylogenetic context, trying to see where's the origin. So we did 2 main phylogenies, one with the chloroplasts and the other with nuclear genes. with the chloroplast trying to get the maternal inheritance of this clade, and for the nuclear phylogeny trying to have more complex, trying to fill the gaps of these more complex stories. For the chloroplast phylogeny, we wrote a program script that can be accessed through the GitHub if you are interested. But basically, the thing that does is download the accession number from GenBank. So the only input that you need is the accession number. So you put the accession number, the program go download the accession, map with a reference, a chloroplast reference, call the variants, and then conduct a consensus lab once you have the variants, and then polish the consensus assembly based in the reference that you are putting with Pylon. That is for the chloroplast, and in some ways is the easy part. Our question was, okay, how we can play with the nuclear genes that we want to capture in some ways, trying to build these phylogenies. And our first idea was to do the same thing. Okay, we have the Nicotiana benthamiana genome. Let's map all the Suaveolentes species to the Nicotiana benthamiana genome and try to call a consensus. But of course, these species are quite different between them. Some of them diverged more than 4 million years ago. So they can be quite different. So first, we have a different mapping rate. So the ones that are closer to benthamiana has a higher mapping rate than the other ones. But the thing that was more worry for us is that you may have regions that are not present in the reference that you may have duplicated in the one that you are trying to rebuild. So for example, here, no? So imagine that you have the target. So in this case, imagine that for the target you have two of these regions, and the problem is that you have only one for two of these regions here, this BP1 and BP2. But in the reference, you have only one because maybe benthamiana only have one of these regions. It has lost the other homologs. The consensus that you are going to call is a chimera. So at some point, we decided that this may not be the best option. So the thing that we did was, okay, so let's try to do some de novo assemblies. Even if the coverage is not good, we may be able to capture the regions that are more conserved and more easy to assemble. For, of course, not the repetitive regions, but maybe the other ones. Okay, so, and we knew that we produce very fragmented assemblies and was computation intensive, but was the option that we decided to use. Okay, so that is the context. We took everything that was public, we took all this resequencing data, and we're starting to do all these different assemblies. We also did the annotation of these assemblies, the functional annotation of the genes and so on. And then we're starting to do some phylogenies. And of course, we're starting with the chloroplasts. So this is a summary of all the different chloroplasts that we reconstruct, not only for the Suaveolentes species, but also for the other Nicotiana species. Let me tell you that it's interesting that even for some species that we didn't have genomic data, the transcriptome, most of the time is contaminated with chloroplasts with enough depth that you can catch the chloroplasts and do the assembly. So for example, for some of these species like Noctiflora, we were able to take all the transcriptomic data to rebuild the chloroplast genome. We ended with, so for some of the species, we still have some gaps because this was a mapping reference guide assembly. So we still have some gaps, but these gaps were more or less small. So we thought that initially it doesn't have a big impact. So we ended with around 54 sequences and 160,000 columns. That is more or less the sizes of all these genomes. It's interesting that we found around 3,334 parsimony informative sites. That for me, let's say that it's a good representation that even if the chloroplasts are big, the part that change from one species to another is not much, okay? And we did the first phylogenies with this RQ3, maximum likelihood method, okay? More or less, all the bootstrapping values were high because we were comparing the whole genome. We have some low bootstrapping values for some of the nodes, but more or less was kind of a solid phylogenetic tree. And our first surprise was that the diploid species that was closer to all the Suaveolentes was Nicotiana sylvestris. And it wasn't what was described, that was what was published. We, of course, repeat this with BIS, a Bayesian method, and we found just the same thing. So, and also, well, we also use the Bayesian method to estimate the divergency date that, or the polyploidization event that was around 5.4 million years ago. Okay, so that was surprising. Say, okay, sylvestris. So why? So because, as I mentioned before, has been studied and is quite solid. A quite solid work was that the maternal donor is Noctiflora. So of course we went back to our chloroplasts and we rescued all these different markers that traditionally have been used to do these phylogenies, and yeah, it was fine. We found that the Noctiflora clade was the one that was closer to the Suaveolentes. Maybe the bootstrapping values was not good for everything, but it was quite solid here. So why? So it was one of our questions that we had at that point. So the thing that we did was, okay, so let's try to break our chloroplasts in the three most blocks, conservative blocks. That could be the inverted repeat. We use just one copy, and of course the short and the long region. And when we did that, we found that with the LSC, we found that of course that it just matched what was known, that is Betinoide, Noctiflora, and Glauca as the closer, the diploid section. Noctiflora section is the closer to the Suaveolentes. The same thing happened with the SSC region. But when we go to the inverted repeat, we ended with the same results with the whole chloroplast genome. And well, doing some, we think that it may be that we may have some chimeric plastoms. It's one possibility, but in order to have chimeric plastoms means that you need to have first a hybrid, but of course we know that Suaveolentes come from a hybrid. The second, of course, you need to have this recombination of the chloroplast, and the thing that is most important is you need to have a biparentally inherit chloroplast genomes, okay? And that it may not be so easy. My surprise was that I found that this publication that say, or that describe, that even if it's not a common process, Nicotiana sylvestris may donate to, when it's producing hybrids, some part of its genome. So even under or concept that is still, even if Noctiflora looks like it's the maternal donor, okay, for the Suaveolentes species, we cannot discard that we have this event that may produce this chimeric chloroplast. That was the thing that we found. We still, of course, we are analyzing this, and we would love to hear about any suggestion or comments that you may have about that. Okay, so that is for the chloroplast. So we went back to the nuclear genome, that of course is the most complicated part in terms of phylogenies because we have a polyploid. So this is in more detail the pipeline that we use. So we use two different approaches. So we try to use two different approaches. One of them was de novo assembly, and the other was just to get this reference mapping assembly, as we did with the chloroplast, with the benthamiana genome. That is the one that it didn't work, as I mentioned before. So this was the one that we tried. We did the assembly, and once we have this de novo DAF, we close the gaps, and then we produce this final. Or sometimes we try to do some rescaffolding using the benthamiana. Just the rescaffolding, the assembly was already done, but we used the rescaffolding. And once we did this rescaffolding using the Nicotiana benthamiana genome, we did another round of gap closing, trying to extend already the contents that we have, and then break it, and then using their own mate pair or pairing information, trying to do the final assembly. And this is our two examples with Nicotiana africana and Nicotiana plexicaudis about how this works. And the problem is that it depending on the genome that you have, depending on the coverage, depending on the complexity, the results are different. because these are very complex genomes. So for example, in the case of Nicotiana africana, when we did the guided assembly, the Busco score was very low, 32%. When we did this with Nicotiana amplexicaulis, we hit 43. So amplexicaulis makes sense because amplexicaulis is closer to Nicotiana benthamiana. So we were able to reconstruct more of the genome. But when we did this de novo and parent scaffolding, we ended with 77. We decided to break these scaffolds and use benthamiana to do a new rescaffolding and then fill the gaps and break again. And we were able to hit a 92% of Busco. That in some ways was the thing that we were looking for, trying to get some, not complete because we cannot have a complete reconstruction of the genomes, but the more complete that we can, okay? And of course, this didn't work with amplexicaulis. Not in this case, the genome assembly, the novel was with a very low Busco. And even if we did the rescaffolding, we didn't improve much. Because initially we were interested only in the origin of the Suaveolentes and not in the diversification process at this stage. We selected only 3 genomes to work with the phylogenies that also will have a decent genome assembly quality. That was Nicotiana africana, Nicotiana cavicola, and Nicotiana debnegi or forsteri. And the thing that we wanted to do is we tried to take the Busco genes and do phylogenies only with Busco. And let me tell you that it didn't work, okay? Because the bootstrapping values for the Busco genes were very low. So the thing that we tried to do at this point was trying to do a gene family approach, just to cluster everything and trying to select single gene families, families that you have only 1 copy per species for the diploids, trying to do the phylogenies here. So we did the gene clustering with MCL, we did sequence alignment and refinements with guidance, substitution model optimization with IQ3, and then we produced the trees with IQ3 again. We filter all the trees that has nodes with bootstrapping values below 70. And then we did this node weighting and consensus tree reconstruction that I'll explain later, and the identification of the homologs, depending of the position. So the problem still is, our idea was, once we have all these phylogenetic trees at the end, we can try to do a consensus, for example, using a tool like Astral, to have a consensus and finally have the consensus tree for the Suaveolentes species. But the problem is that Astral is not designed for polyploids. It cannot handle well the polyploids, and of course you have some problems. So you need to do some homologs identification first. So the thing that we did was we analyzed all these trees first. We did some ancestral node analysis, and then we identified the homologs. And then once we identified the homologs, we rename the different leaves in this case, and we did the consensus phylogeny. To do that, we did a pipeline that you can find also in GitHub with different modules. So all these things were in this pipeline. And of course, the first step is to do the gene families. Gene families analysis are not easy because you can use different parameters and you need to optimize those. So the thing that we did was when you are doing the gene cluster, there is a parameter that is the inflation value that in some ways tells you how big is going to be the groups or how small. So you can increase the inflation value and you can split the number of clusters. And the idea was that we run different ones, different values, until we maximize the number of single copy genes per species in all the clusters in some ways. So this is the thing that is representing here. And we ended with that, our inflation value in our case was 2. 2 was a good number. That was a good balance between having a good number of clusters and also single copy genes. Then the next steps was doing the sequence alignment. And of course, doing 1 or 2 sequence alignment when you can supervise is okay, but when you need to do thousands of them, we want to try to maximize that. So in this case, we use guidance that is kind of a program. I really like it that it can improve, it can polish automatically some of these alignments. So here is an example. Now in red are the bad alignments of these species, and after this program ran this processing pipeline, some of these scores can improve. So we used guidance to optimize the sequence alignment. We also needed the substitution model is important. So we use IQ3 to estimate the substitution model. So we put that in our pipeline. And we ended that there is two majorities of substitution models that was the ones that according IQ3 were the best. So what are the ones that we use? And we ended with a lot of different lists. So the first problem, as I mentioned before, is that the polyploids cannot be used with Astral, no? Because if we use this topology and we have, for example, here Nicotiana benthamiana and here Nicotiana benthamiana, we try to call a consensus of different trees, Astral is going to give us a problem. So we ended with this idea of first, we need to mark the specific ancestors. So for example, in this case, if this Nicotiana benthamiana gene or leaf is close to the Silvestris, this will be a Silvestri ancestor, or we will mark as Silvestris. In this case, we have this Nicotiana benthamiana that is closest to the Noctiflorae clade. So we will mark as a Noctiflorae and so on. So the thing that we did is we're starting to mark and trying in our pipeline, trying to identify these genes. The next thing that we did, of course, in the process to do that is we're starting to define these nodes to say, okay, so under this node, because the sections are, in most of the cases are monophyletic, all the sections are monophyletic. We're starting to say, okay, so under this node, this is the node in which we have, for example, Suaveolentes and Alatae. Under this node, for example, here, no, Suaveolentes and Silvestris, and here we have Alatae, so Suaveolentes and Silvestris. So we're starting to go and annotate all these different nodes because we needed to identify the nodes in order to identify the homologs in our script. And then we pop up with the idea that if we just count nodes, maybe if we count nodes, we are going to be able to infer some phylogenetic relationship. Remember that this are trees that all the nodes have been supported by 70 or more in the bootstrapping values. So in theory are very well supported. So we're starting to mark these nodes to infer their origin. So we do them, and we're starting to count them. And for example, in this case, in this example that I showed you, in this case, for example, we have two nodes that are Alatae Suaveolentes, and we have 3 nodes that are Noctiflorae Suaveolentes. So for example, in this case, it's indicating that the two ancestors of Suaveolentes potentially could be Alatae and Noctiflorae. This is the thing that we did with this tool that is this multi-tree analyzer. So we did with everything, no? This is the summary of all these different processes. So we ended with 51,000 clusters initially, but when we started to select only the cluster that with some sizes in order to have only one copy for the diploid genes, we ended with 2,000. We did the sequence alignment with guidance, the substitution model optimization, the tree production. We ended with 1,633 trees. Then we did the node filtering with the bootstrapping values more than 70. We ended with around 931 filtered trees. And then we started to do this node weighting and the tree construction, okay? And this is, we normalize the nodes depending on how many leaves do you have behind. So this is not just the number of nodes, but we counted the node about that. And of course, the sections that are monophyletic, you are going to have a very strong numbers, very high numbers, because all the nodes are going to say, okay, this is monophyletic, as you can see for the Alatae, Noctiflorae, Pitinoides, so that makes sense. The thing that was more interesting is that we're starting to find that you have a lot of nodes, that is Alatae Suaveolentes. Of course, you have a lot of nodes that are Nicotiana Silvestris or Nicotiana Tomentosae, because we know that Nicotiana tabacum derived from these two. But also we found the ones that are associated to the Suaveolentes, as I said, as Alatae Suaveolentes, Noctiflorae Suaveolentes, Pitinoides Suaveolentes, and Nicotiana Suaveolentes Silvestris. So if we represent this into a tree, these numbers, this is the thing that you can see. So as was described before, Noctiflorae and Suaveolentes, you find a lot of trees that support that Noctiflorae is one of the progenitors. And you find some, a little bit less, that also support the idea that could be Pitinoides. Interestingly, we find also a lot of clades in which the Suaveolentes clade is sister to Petunoides and Noctiflorae. Okay, we found this number that is higher. In the case of the other one, we found a very, very strong number of nodes, very strong value, a high value, sorry, that say that Suaveolentes are with the Latae clade. Sometimes also we find with the Silvestris, and then the rest. No? So as I mentioned before, we use as a control the Nicotiana, that is very well supported. So according to this, just the node weighting, one of the ancestors will be Petunoide Noctiflorae, as was mentioned before, and the other will be the Latae Silvestris, probably. Nothing new. Of course, once we identified the homologs, we went to the benthamiana genome, and we're starting to color our benthamiana genome with chromosomes depending of the ancestor. The thing that is interesting, for example, here also is that you can see a lot of red because it's the Latae origin. You can find a lot of blue that also you can find that is the Silvestris origin. Noctiflorae you can find also. Petunoides not so much. Okay, just some small number of genes. And the thing that is interesting and for us was reassuring is that some chromosomes are enriched in some of these origins. So for example, chromosome 9 is enriched in the Silvestris origin. Meanwhile, for example, chromosome 6 here is enriched in the Latae origin. Remember that this has been done with 411 genes. It's not the whole genome, but the genes that we use for this gene cluster that we use for this analysis. When we started to have all these 411 different trees, we're starting to classify if, depending of the origin of the Suaveolentes, it was Latae, Noctiflorae, Petunoides, Silvestris, Noctiflorae, Petunoides, and so on. So we classify all these different trees. The one that, for example, was more common was the topology in which Suaveolentes was Latae and Suaveolentes also was Noctiflorae. And we did one astral tree for each of those. Okay, just trying to have what was the final reconstruction of those. So this is an example for the type 1. Okay, so in the type 1 of the astral tree, we can find here all the Suaveolentes species that are close to Latae. In this case, that is not doesn't appear Latae as a monophyletic, because this is the consensus, but in the consensus, in some of the trees, of course, the Latae are the monophyletic sister species to Suaveolentes, okay? And in this case, for example, we have here the Suaveolentes sister to Noctiflorae. The thing that was interesting is that in some cases, on some of these trees, we found that sometimes one of the species, it wasn't in agreement with the other ones for the Suaveolentes origin. So what means that? Here we have an example, no? The type 14 with 23 trees, no, it's seven nuclear trees. We have all these Nicotiana Suaveolentes gene cluster that is the Negia africana, benthamiana, and clavicola. Africana sometimes, most of the time appears as external to the Negia, but in this case appears as internal. Here we have also, remember these are consensus trees. These are associated with the Silvestris and tabacum. But for some kind of reason, in this case, we have the Negi that is associated also to Latae. And when we are starting to look for these trees, we found trees in which you have maybe all the Suaveolentes that comes with Silvestris, the other Suaveolentes come with Noctiflorae, but maybe one of the Negi goes with Latae and not with Silvestris. Meaning that maybe we have a different fractionation or a different origin. We still don't know. We are still discussing that. That was interesting. Of course, we wanted to date this, no? And we found that they derive from Noctiflorae, Petunoides, Latae, and Silvestris. But Noctiflorae and Petunoides was the question about, okay, derives from one of them or both, a hybrid of them, So we ended with the question that, of course, we need to date the diploid divergency. So we took all the 411 single copy genes without the polyploids, and we're starting to date the divergency for all of them. So this is the consensus tree with BIST, and we found that Noctiflora is sister to Petunoides, and they diverged around 5 million years ago. And then Alata is sister to Sylvestris, and they diverge a little bit older, so has a lot older divergency. Then we did the same dating with all the different trees that we have for these 411 trees with all the Suaveolentes and Nicotianas, and then we plot the dates. And the thing that we found is that in green you can see the divergency dates for the split, for example, between Alatae and Suaveolentes, Sylvestris and Suaveolentes, Noctiflorae and Suaveolentes, and Petunoides and Suaveolentes, and you can see that it's around 5 million years ago. But when we look, for example, the split between Alatae and Sylvestris, it overlaps with this split. It may happen just a little bit before or a little bit after. Our hypothesis was that this is a little bit before, because we don't find many trees in which Suaveolentes is sister to Alatae and Sylvestris at the same time. But we found a lot of trees that Suaveolentes is sister to Noctiflora and Petunoides, and indeed when we date the split between these two diploid sections is younger than with the species. So we think that probably one of the ancestors is not Noctiflorae or Petunoides, but is the ancestor of those. Okay, we think, our hypothesis is that is the ancestor of those. So if we just changed colors again, just thinking that we don't have Noctiflorae or Petunoides, but maybe the ancestor, this is how we look like the chromosome painting. Okay, so just the conclusions, just wrapping up. We have developed this Nicotiana benthamiana genome. 95% of the genome is encoded to 19 chromosomes, 61,000 genes, more or less. The quality is okay. Then the Suaveolentes section, we think, or at least our results indicate that come from a polyploidization event between the ancestor of the Noctiflora and Petunoides clade. And we don't know still if it's a hybrid between the Alatae and Sylvestris, or maybe it's also the ancestor too. But the thing that we know, now that we have been using a lot of Alatae genomic data, is that the Alatae is has an important contribution in the Suaveolentes genomes. We think that the Noctiflora Petunoides ancestor may be the maternal donor, according to the whole Chloroplast genome phylogeny, but also we found some evidence of chimeric chloroplasts, we think. And also, just looking into all these different trees, of course, we are still analyzing the trees, but analyzing these trees, we found that different Suaveolentes species conserve different homologs, reflecting maybe they have different early evolutionary path. We still don't know. We want to, of course, to complete that, but we still don't know. Okay, beside that, I would like to thank all the people that was involved in the Nicotiana benthamiana reference genomes, as you can see here, especially to Brian Kipko and Celine, that were the main donors that give us the data to be able to jump to chromosomes. And then all the study of the origin of the Solanaceae sections, all the genome size measure was done by Elia and Thomas when I was at Virginia Tech. Some of the phylogenetic analysis was done by Lucio and Lea when I was in the University of Milan. And of course, very big thanks to our collaborators, Nikolai and Nicolas from Philip Morris that fund part of the project and of course, deliver also some of the data that we analyze. Thank you very much.
Thank you, Aureliano. That was a great work and great talk. Yes, it's a lot of study and a lot of data there to disentangle the origins of Nicotiana benthamiana, Nicotiana genus in general. So now we are going to start with questions. So remember that you can add your questions in the chat or just type question and I'll give you the word. So first we have a question of Luis Augusto Caos dos Santos.
Hello, everybody. So thank you so much for the really nice presentation, Aureliano. It's really nice to see all the progress in your work with Suaveolentes. And we are also benefiting a lot for this progress here in Vienna. So it's really, really nice. I have some questions about some about the analysis and also some about the findings. So first, regarding the chloroplasts, it was really interesting to see these inverted copy regions being quite sharing all this identity with the paternal chloroplast. So did you saw other types of rearrangements between the chloroplasts that you assembled for the section, or you saw just the inverted region that shows this possibly common identity with the paternal?
Um, well, I mean, the first analysis that we did, of course, was just to split in these 4 regions, because it's kind of the easy from concept. But then we pop up with the idea, and I think that I don't know if Lucio is here, but of just not to divide, but just to scan. No, say, okay, let's take I don't know, 1,000 or 5,000 base pairs and do a phylogeny, and then another, and do another one, and then scan it. So he just he finished several months ago a pipeline to do that, and we were proving that pipeline with Solanum and worked it pretty well. And then he's doing that now with Nicotiana. But we decided to, if at some point we produce something, we wanted to not include it here, but later, because this is something that we are trying to do the pipeline first, and then trying to see how can fit with the analysis, because we wanted to add we have other questions for the pipeline too. Okay. But yeah, you are interested in this kind of analysis. I think that probably Lucio, I can put you in contact with him. That is the one that produced this scan.
Yeah. A combination between different chloroplasts are really, really hard. And normally, some of the regions of the chloroplasts also start to increase in repeat content. So then, yeah, so the recombination can maybe occur, but I think it's still quite hard to have like hybrid and chimeric chloroplasts.
Yeah, and indeed, I'm not sure if someone has been able to reproduce a chimeric chloroplast somewhere. So the thing that we have been reading is that people hypothesize that it's possible. But
yeah, there is a nice paper from a group from Max Planck that they work a lot with chloroplast genomics, and I think that they tried in some of the groups, but I don't know if it worked at well that.
Yeah. Yeah, it's the reason also why we thought that maybe just to divide with inverted repeats and the rest of the chloroplast makes sense in that sense, no? Because when you are reproducing, you know, is the structure. Yeah. But still, don't know. The thing, in some ways, the thing that we were trying to answer is why, why our chloroplast genomes doesn't give us the results that everyone was obtaining for years. No, what is the thing that we were doing wrong? No, so and that is the thing that we found, that when we divided in these sections, they give us different information.
This would explain all the incongruencies, what we saw in the past. So I found really, really interesting this result from Debny and Africana. And I would ask if you look at for some patterns of introgression between these two species with this data.
Yeah, we are in the process. As I said, we have all these, we are still processing all these genomes. And yeah, the next step, of course, is just to look for integrations with the whole genome assemblies, and also just looking for genes that may be, we are not only interested in the, also in patterns of integration, but also different patterns of fractionation.
Yeah.
That may have, not because you have, because we have a lot of different combinations. Maybe we have 2 different polyploid origins that then starting to integrate between them. Maybe we have one single one, but that the fractionation at the early stages of the evolution of these species were very different. we still don't know. But of course, I mean, yeah, it's something that we want to look. As I said, initially we were more interested in the size of the parents, no?
Yeah. So now you are going more in the other species on the section. It's the second part of the aim.
Yeah, in some ways we have all this resequencing of these genomes. Still, I can say that, I mean, Illumina still doesn't give you high quality genomes in that sense. We are able to recover genes for sure, but maybe not all the genes that we want. Maybe we can evaluate repetitive contents too. But again, no, I mean, of course, I'm summarizing here this, but I also have been measuring K-mer distribution, trying to do phylogenesis with K-mer distribution. That was a complete disaster initially, and all other methodologies. But yeah, we are still trying to characterize those, no?
Did you had a lot of impact of missing data for these phylogenies? Because I'm working with the HAD part of in Suaveolentes, and even if I got like a high number of variants at the end, I always have like some regions of the tree that I don't have like really high bootstrap. So, and this is really impacted by missing data. So I wonder if also this happens when you use this approach.
Well, in this case, we don't have missing data because we are starting to work with gene clusters. So when the gene cluster, the requirement was that we need to have everything. No, we need to have everything. We need to have everything. So we have 411 genes in which we have one copy of each of the diploids and 2 copies in most of the cases of each of the polyploids.
Okay. Congratulations again. It's a really nice work, and yeah, I really enjoyed your presentation. Thank you.
Thanks.
Thank you, Luis Augusto. Then we have another question from Federico Roda.
Hi, Aureliano. Thanks a lot for your talk. Interesting. I have a question which you started answering before, but is the data good enough to do functional analysis? So can you, for example, answer questions about the importance of polyploidization in the evolution of traits like nicotine production or any other trait? Or you probably don't have long enough sequences or enough representation of every gene in every sample?
Well, I think that usually functional analysis are better done when you have populations rather than different species. So just comparing different species, I think that is a long shot. So, I mean, we didn't start to look for specific genes here. For some of the species, we have almost full length genes. For others, we have fragmented genes. But no, I think that is a long shot in this case. I mean, especially, I can say also from my point of view that that we don't have a really good phenotypic characterization of this species. I mean, in terms of, Mark, of course, and his team have been doing a really good recollection and phenotyping of this species on the wild. But I'm wondering, or at least from my point of view, working in crops, I think that you need to put everything in a greenhouse, put some stimuli, and then starting to do some experiments there in order to being able to relate the function of the genes with some of these genome architectures.
Okay, thank you very much.
Thank you, Fede. I don't know if there is any other question. Gustavo Silva, yes.
Hi, Aureliano. Nice to see you, and yeah, very nice talk. Thank you for sharing all these results. I have a little bit of curiosity of that result that you showed in the distribution in the chromosomes of the different blocks that could be, for example, enriched by one specific antecessor And I'm wondering if you already started some analysis, for example, of the structural dynamics in the genomes to try to explain a little bit why, for example, yeah, some chromosomes could be enriched by specific antecessors that maybe it's like, I don't know, preventing some kind of introgression from the others, or these kind of dynamics, for example, the inversions or some specific parts of the genome that could be more resistant to bring genomes from other species or something like that.
No, not at all. We still didn't started to do that. As one of our plans, of course, is once we have a decent assembly of those, that is the thing that, of course, is the most time consuming with this data. It's time to see present absence of gene families and see, for example, our idea here is just to more than characterize the story of the Suaveolentes, because we don't have the resources to do it, and because I think that you really need to have a good population to do this. We are more interested to see how once you have the polyploidization event, how the fractionation of the polyploids works just with present absence of genes. If you have gene families that still keep all the copies, and we have gene families that come back to the diploid state very, very fast. We are interested to do that in general terms, but of course, the evolutionary trajectory of each of these species is very, very different. No? Benthamiana is very different of Nicotiana maritima, for example. So I think that I would find difficult to associate, as I said, some phenotypes, some characters, and the genome dynamics to those.
But for example, using these like these patterns of the synthetic blocks, you cannot derive some, like at least some rough conclusions about some specific regions that could be, I don't know, more tend to be more conservative.
Yeah, so that is a good question always for polyploids. When you keep synteny and you keep two homologs, is a stochastic process or is selection? And I guess that the problem is that, yeah, you need to have enough number of representatives to go through that. So it's always my question, no? So yeah, I don't know if, as I said, when you say, yeah, we keep here this synteny, of course you have, I mean, the number of recombinations that you may have between the two homologs in, let's say, 4 million years is a lot. So in theory, maybe the recombination block should be relatively small. But still you can find synteny between the Suaveolentes species and Nicotiana tabacum or Nicotiana sylvestris. So yeah, I'm still not sure. This is this kind of question that I try to approach carefully. Yeah.
Yeah. Okay. Thank you.
And that's let's think that is supposing that your genome assemblies is okay. So I'm still. I mean, this genome assembly is fine, but of course we have some errors. So yeah.
Yeah. That's true. Thank you.
Yeah.
Very interesting.
Yeah. Thank you.
Thank you, Gustavo. So now it seems that we don't have any more questions. So with that, we are finishing. Oh no, it's only Carolina Turchetto that says, thank you, Aureliano. It was a really nice talk. Very several congratulations. So yeah, it was a great talk. With that, we are finishing this seminar today. And in 2 weeks, we are meeting again for the talk of Xia He. And he is going to talk about establishing 2 physalis genomes as new Solanaceae systems enables genetic reevaluation of the inflated calyx syndrome. With that, thank you everyone, and see you in 2 weeks.
Thank you. Thank you very much.